Hi, everyone. Welcome to the 8th ActinoBase e-seminar. Thank you very much for tuning in. Just to begin with a few brief reminders. We are still accepting abstract submissions for talks for e-seminars from both PIs and postdocs and PhD students, if anyone would still like to give a talk. We are also accepting submissions for flash presentations for our ISBA special next week on the 25th of June. So these will be short 5-minute presentations. If you'd like to submit your abstract for that, the link should be in the most recent email from ActinoBase. And the deadline is going to be midday Monday for submissions for that. So please do submit an abstract if you're interested. Today, the format of the talk is slightly different. We have the first of our group leader talks. This will be from Dr. Virginia Helena Albarracín from the Center for Electron Microscopy at the National University of Tucumán in Argentina. So I will pass you over to her now, and please remember you can ask questions in the YouTube chat box and also on Twitter using the hashtag ActinoBase08. So Virginia, if you'd like to start sharing your screen.
Okay, thank you. Thank you very much. I will start there. Okay, so good afternoon to all of you. I'm very happy to present our work from Argentina to the world in this ActinoBase seminar, which I think is a very interesting initiative. I would recommend it for all, so to present here. I will talk about the work we have been doing in Tucumán. Tucumán is in the northwest of Argentina. I am the head and group leader of the Center of Electron Microscopy, but also I collaborate in this project with the bacteria from High-Altitude Andean Lakes with a group of another institute in Tucumán, which is proteomic, and the lab of research on Andean Lakes, Linda. So I will make a brief outline of what I'm going to try to show you today. First, I will talk about the environment where we isolated and studied its trends, which we call High-Altitude Andean Lakes. After, I will talk about what are the findings after exploring all this area, this fascinating landscape and pristine environment, about the extremophile biodiversity, and especially about Actinobacteria, which is mostly my subject of interest, and of course this seminar. And within this Actinobacteria, we use them as model for study the response to or the adaptation to a stress, which is UV radiation. And we call this system of adaptation as UV resistance, and I will explain a little bit later. Okay, first, the High-Altitude Andean Lake. I told you these are environments, pristine environments from the Central Andean area in South America. It covers part of Argentina, Chile, Bolivia. And in this area, which is very extreme because it's a high altitude above 4,000 meters, you have mostly desertic and arid conditions. And in between, you will have a lot of lakes, shallow lakes, distant from each other. These are only a few of the ones that the group in proteomic of Dr. Farias has sampled for already 20 years. These are only from Argentina, the ones that I'm showing here, but we also have explored the ones in Chile. And there are also groups working on all the microbiology of this environment. So why it's important, or why we are interested in study this system? Well, because they are considered extreme environments. And one of the things that are more surprising is that as there is a high altitude and a low latitude, this area, the Puna region, and of course within them, the high-altitude Andean lakes, are exposed to high levels of total solar irradiation, but also of UV irradiation. So here you can see a map of the mean daily total solar irradiance and the in one side, and the UV index in the other side. And this is a global analysis made with data adapted from NASA. Here are the areas, the stars correspond to the lakes that were sampled, and that we have microorganisms or DNA from there. And as you can see, as it is yellow or orange, it is higher the UV index. And also the total solar irradiance is higher and is the highest of the world. So if we have here the highest irradiation on the world, the microbes that are living there must have systems to allow them to survive to this pressure. But this is not the only pressure they are enduring. They also have a lot of other extreme factors that they have to take care of. And as you can see, we are interested in the microbiology because apparently there are no life in this area because it's a desertic, arid region. You will not find too much animals or birds or reptiles or even fishes. You will find only tiny microbes like Actinobacteria. So, and that's why, because the high UV index is one reason, but also there are low oxygen pressures due to the high altitude. There are drastic daily temperature change because it is so high. When the sun comes down, it becomes very cold immediately. Another thing is the chemical stress they are suffering. This area is from the geological processes origin from the earth. They have a lot of arsenic that is in the soil, but is also in the water, up to 200 ppm. Take in mind that arsenic content in water for consumption for human is less than 0.001 ppm. So this is a really deadly arsenic concentration. And also these lakes, especially the lakes, are hypersaline. Some of the lakes are acidic, or most of them are alkaline lakes. So altogether, the microbes that live here, they have to attend all these conditions. So we call them as polyextremophiles. And these polyextremophiles were sampled in different niches. One was the lake itself, in the shallow water from the bacterioplankton, but also from the bentos and from the soil around the lake. In some lakes, you will see these mats that are forming. So also the group has sampled these microbial mats. And interesting is that there are, I said to you that there are very little large animals, but one of them is the flamingos that disperse the microbial from one lake to the other. And we have also studied the flamingo feces for that microbial. lately, in the last 10 years, the group has shifted the interest to these kind of structures, which are called and studied hopefully by Dr. Faria's group as the ecosystems, microbial ecosystems associated with minerals. These are special kind of microbial mud that over the time has sediment and mineralized that forming which are called as microbial. These microbiologists can have different shapes and forms. One of them is the stromatolites, which are this kind of stromatolites. These are from the Socompa Lake at 3,600 meters above sea level in the province of Salta, in the northwest of Argentina. And these are layer systems that have microbes in mats in different layers, but they also have minerals and constitute like living rocks somehow. And why they are very important is because the first forms of life on Earth were stromatolites. And when you have a modern stromatolite that is growing, and actively growing, you can use this as modern analogs for the Precambrian ones. So you can study the evolution of the life in these small structures. After I show you only if you are interested, you can read all of this about the diversity of polyextremophiles from Hall in all these papers, specifically about all these ecosystems associated with minerals like the stromatolites, the microbials, and all of them. Here are two reviews about this, and there is also a recent publication on all the systems that are microbial ecosystems that are surviving under these conditions in the Central Andes region. But I think I have convinced you that we have a high microbiodiversity in this area, even though we have extreme conditions, and even though we have the highest UV radiation on the world. So there must be something that make these microbes to thrive in these conditions. And so there are molecular and physiological mechanisms that we call them as UV resistors. We start exploring the UV resistome already in this paper in Gamma Proteobacterium, Acinetobacter ver3, isolated from Lake Verde. Now I will show you how we follow this in one Actinobacteria, which is Meresthencon. But the initial work, the initial idea was that there was this UV resistome that have a different subsystem. One was the microbe to resist this UV has to adapt to it in different forms. One is with the sensing and response regulators. So the cell or the microbe must see this somehow they have to sense the condition that they are exposed to, and in this, when they sense it, they can respond fast or effectively. And how they respond, there are different ways. One should be like avoiding or shielding, so they can escape, for instance, using system of locomotions, or they can protect themselves if they cannot move, which is the case for most of the microbes, and especially in these lakes that they don't have enough natural shielding. So they have to use these pigments, stremolites, or other metabolites to protect themselves from filter somehow the UVB that is being produced. Well, if this is not working, then they have to generate a fission stress response so they can neutralize the effects of the oxidative damage and the reactive oxidation species. If the system of response is not enough, then they have to have a high tolerance to damage. So they have to follow or to continue working, even if they have damage to the biomolecules like DNA or proteins or lipids. So this has to be another very important system. And if the system DNA is damaged by photoproducts produced by the UV, they have to have efficient repairing system that we divided in the ones that need light, like the photolysis, or the ones that don't need light for work, so dark repair. So this was the general hypothesis that we have about this UV resistor. And of course, I'm not working alone in this, apart from the sampling and all the characterization of the environment that is done by Dr. Farias and her group in PROIMI. What we do is that we have a group that works in light sensing, in the system of light sensing, and I have one PhD student that already gave his thesis, Luciano, who works on the photoreceptors of some of the isolated, including Actinobacteria. Daniel, which is working from the metagenomics point, digging in all the sequence that we get from this environment. And then we think of studying the response. The response, there are two students, PhD students, Federico and Natalia, that are working in these mechanisms of resistance, trying to integrate all the conditions or the molecular system that we see here. Silvina and Cecilia that are working in how they, if they have a response, they may have communication between each other to help this response to do it more in a massive way or more active. And of course, because these microbes come from a very extreme environment and little study, we are thinking about applications, and that's how our postdoc Anaí is working on this already, and Cecilia also, which is a postdoc too. How we study this UV resistance, where we have different approaches. We can study all the system, the molecular system, directly from the DNA we take from the sample in the field, or we can isolate some model extremophiles and start doing physiological experiments or multiomics approach. And finally, this is the line I was working in the last 8 years already, which is studying in particular the photoreceptors, so the ones that can detect the light, and the ones that can repair DNA, which are the photolyases or cryptophores. Now I will talk more about the UV-resistant Actinobacteria that we found there. This is the paper where we describe and I made new isolations. Some of these isolates or strains were already described in previous work. So what we do, we make a general screening, taking the new isolates and the strains that we already have in the lab from former expeditions. And we make a profile of the polyextremophilic capabilities under chemical or physical stress, and also we characterize them by microscopic technique and phylogenetic analysis. We found a rich diversity of Actinobacteria especially the rare Actinobacteria, like Micrococcus, Microbacterium, Nesterenconia, Cocuria, Rhodococcus, Artobacter, Micromonospora, Blastococcus, Brevibacterium, and Citrococcus. We took, some of them were isolated from the stromatolite. For instance, we have a Micromonospora from the stromatolite, which was very interesting to find there. But we used as a model Nesterenconia because when we started all this project, there were very few studies on this genus, and we wanted to go further because we know that in general Nesterenconia, where all the microbes are very resistant to arid conditions, they are allophiles, so we continue studying this. So what we did first was to identify by molecular methods what type of Actinobacteria possess. So we compare it with the described Nesterenconia, and we found that it's very near to the clade of Nesterenconia allotolerans. This was made by 16S, and this is a whole genome analysis with average nucleotide identity. And as you can see, Nesterenconia is forming a different clade. This is the macroscopic aspect of the strain, and this is how you see it in a scanning electron microscopy. Then we characterize it for the UV resistance, and we compare it with the closest taxonomic strain, which is Nesterenconia allotolerans. This is a very easy essay. This is spots of a diluted culture of Act20 and Nesterenconia allotolerans. This is without exposure to UV, so this is a control. They both grow well in this media, but when we apply a certain dose of UV, then we see that Act20 is much more resistant. Nesterenconia allotolerans is a resistant strain. In general, I have said that Nesterenconia are resistant to a lot of extreme factors, but yet it is less resistant than Actinobacteria, which was isolated from a highly irradiated virus. And this is the UV resistant profile when we apply different doses of UV, so after 2 hours and so on. Yes, and we can see that still at 20 is even the highest dose that we test here is highly resistant. We also try to see how if we can observe changes in the morphology or in the ultrastructure of Nesterenconia by electron microscopy. And this is the same essay I showed you before, but instead of doing checking the growth, we directly make samples for electron microscopy. And what we show, this is the Nesterenconia allotolerans without and with the UV treatment, and this is Act20 without and with the treatment. So it is clear here that Nesterenconia allotolerans is affecting its structure, its morphology, and in the case of Act20, after the same dose, there are no morphological changes. So this is another proof of the resistance of this. The other essay that we made was to check UVB survival in liquid media, but after we see, this is the control, and then we measure the growth after a certain dose of UV, 20 minutes. And this culture that was exposed to UV, we separated and made two different experiments. One we exposed to dark repair, and the other we left it in the light, so it can recover it, and using, if they have photolyase, they were going to recover in this condition. So as you see, both strains, one is F20, this one, and this is allotolerance, they both can recover after UV stress, and they recover much more in photo repair than in dark repair. But we are only here seeing the growth. We don't know what is happening inside of cells. What we do is, in collaboration with Thierry Duqui and Jean Cadet in Grenoble, in France, we had checked about the content of photoproducts that are accumulated in the DNA of all these treatments. So in the control, in the UV, in the dark, and in the dark repair, and in the photo repair, okay, in both strains. So the first indication of the first result of this experiment is that UV F20 have much less damage, so it accumulates much less damage in the DNA than allotolerance. So allotolerance, although the growth was not very different, allotolerance has almost double the photoproducts in this after UV, 20 minutes of UV. So this is the first thing. The second thing is that the dark repair and the photo repair are efficient in allotolerance to deplete the damage. Okay, so we have here less DNA photoproducts, and in photo repair much less. And also is the case for Act20, but dark repair is not much as efficient as in photo repair. These are total photoproducts. That means we are checking the photoproducts that are CPD lesions and the ones that have different chemistry, which is 6,4 pyrimidine, pyrimidone, and DWAR products. So all these are photoproducts, and we have sum up and analyzed them here. But in this case, we have only described, for you to understand better the mechanism, one type of damage, which is a CPD that is made between a T and a C. And in this case, this is a 6,4 pyrimidine, pyrimidone photoproduct between a T and a C. Okay, and here you can see that after dark repair, Act20 can deplete this photoproduct, but is much more defined, or it is lower in photo repair. And allotolerance is not as efficient as Act20. But so this means that they have an enzyme that catalyzes the elimination or the depletion of CPD photoproducts, which is a CPD photolyase. In this essay, we found that Act20 can also deplete 6,4 photoproducts, and this is very important because it is indicating that they must have another type of photolyase that are these 6,4. So photolyases are very specific, and one only use attack or diminish CPDs, and the other only 6,4. Okay, so in this case, in Heteronconia, we have both. So we then, with all this information, we went to the genome to try to understand better this UV resistance. So here is this table is to compare the characteristic of the ACT20 genome with respect to the other genomes that are published. In general, they have ACT20 have the average size of the Nephrenconia around 3, average GC content, and the average coding DNA sequence. With this information, we compare the genes that may be part of this UV resistance. So in this, you will see in different colors the genes responsible for the repairing of DNA, such as base excision repair, nucleotide excision repair, and so on. The subsystem of genes that may be included in the oxidative stress response, the ones that are maybe taking care of the photoprotection of the shielding that I tell you before, which is carotenoids and ectoin, and also these ones that may be included or in the locomotion systems that may allow somehow to protect the bacteria. And very interesting, we found these genes which are coding for the gas vesicles. The gas vesicles are protein systems that help aquatic microbes to go up and down and change in a water column, their position in a water column. So this is a soil bacteria, Actinobacteria. So it's very interesting that maybe it was in one evolutionary, it was in the first place, it was aquatic bacteria, or maybe because these areas are flooding all the time, they are shrinking the water. So maybe in some moments they have water and they need these conditions. Or maybe these vesicles have evolved in some way to protect them for the UV, because as we will see in a few minutes, they are overexpressed in the protein. In the genome, we look specifically for these photolysis. So the previous experiment show us that Nephrenconia have two types of repair under light, which was a CPD repair and a 6,4 pyrimidine repair. So that means that it has at least these two photolyases. And indeed, what we have found is that Nephrenconia have a CPD3-like gene and another gene for related photolyasis that belongs to a nucleate called bacterial photolyase-like protein, which has associated a iron sulfur cluster in its structure. So this was very interesting because by the time when we found it, there was not too much information on this clade. Now they are more studied, they have crystallized some of them, 2 of them, and they found also that this that was originally described as a cryptochrome, this means that they didn't know what they was doing, the function, they found that it has a 6,4 photo repairing activity. So maybe this is the protein that is helping Nephrenconia to repair the photoproducts as we had seen in the experiment. Well, and then we performed the proteomic assay of ACT20 in 4 conditions, similar to the studies that I showed you before. So what we do is we put a liquid culture under UV one of these, after we put a culture under UV, we left, we divided in two, and one is left in the dark for 2 hours, and the other is left in the light, sorry, and the other is left in the dark. And this we call dark repair treatment, and this is photo repair treatment. And this is the UV treatment. And also, of course, we have a control that has overcome all the treatment, but without exposure or change. It was always kept in the dark. Here is a table analyzing the overall results. We have made two analyses. One is qualitative, like this one, and the other is quantitative. In the qualitative, we only take care of if the proteins are present or absent, okay? So what we did is we have the predicted proteome. This is taken from the genome that we have, and this is the experimental proteome. The experimental proteome are all the proteins that were summed up from each of the conditions. So all the different proteins that were found in each proteome, okay? So the predicted proteome was 2,689 proteins, but the experimental proteome was almost 1,600. That means that we have this obtained 60% of the predicted proteome under the experimental condition testing. If you see in this detail, these are the proteins that are in each condition: dark, like the control, UV, dark repair, or photo repair. The treatment with most proteins is the one with the photo repair. It means that after light, after UV, and after light repairing, the cell has somehow mobilized or used more proteins than in the other conditions. This maybe looks small, only 60%, but when we go to identify the pathways and modules from the predicted proteome and the experimental proteome, we see that we have obtained almost all the modules and pathways that are originally in the predicted proteome. So I think this is a very good coverage of what is really happening in the cell. The second part of this qualitative analysis was to determine which proteins are in which system. So we know that there are proteins that are associated with the metabolons of carbohydrates. So we show it here. The ones that are related with amino acids metabolism, cofactors and vitamins, energy metabolism, other amino acids, lipids, nucleotides, terpenoids and polyketides and aromatic compounds. The important thing is that most proteins are conserved among treatments. So if we compare the predicted, the experimental proteome in all these conditions, so in photo repair, dark repair, UV, and the total dark, we will see that in general they are similar. For instance, carbohydrates, the percent of shared proteins among all treatments is 91%. So 91% of the proteins are present in all the condition without mattering which condition was. So 91% of that is present. Which is the one that is most changing metabolism is the one for terpenates and polyketides, which I found this interesting because maybe the secondary metabolite is then used to overcome somehow or this UV stress or challenge. In this slide, I want to show you a detail of only one of the subsystems that we are trying to integrate in the whole UV resistome, which is the DNA repair, which obviously is one of the most important. Because once the damage is produced, it is being repaired and may overcome the stress UV and may allow the survival of the cell. So here between this, in the DNA repair subsystem, we have the different systems that may be integrating DNA repair, and of course are the ones in the DNA replication base excision repair, nucleotide excision repair, mismatch repair, homologous recombination, and photolyase. In these three, NER, mismatch repair, and DNA replication, they are similarly important in all treatments. So they are most the same active in all conditions. But you can finally easily see that the homologous recombination pathway is more active during the, or is changing during this UV photo repair dark treatment. And also a very interesting fact that is somehow supporting our previous finding is that under dark repair, in the dark, that means the cells that were not exposed to UV, they are only expressing one photolyase, so the CPD3 photolyase. That may be basal expression. That this happens exactly the same in the dark repair. It only expresses one photolyase, which is the CPD3. But during the UV and during the photo repair, there are two photolyase working. That means that because there is more damage and because there is light that it is used by the enzyme, they are working, both enzymes are working, or somehow this other protein that I show you, this cryptochrome, that may be a 6,4 photolyase, is helping the CPD3 photolyase to recover from the damage. That was the qualitative analysis, and this I will talk briefly about the quantitative analysis, because we can only also say how much, or if they were proteins that were upregulated or downregulated in some conditions. In this first chart, you see the UV experiment compared with the non-exposed treatment. In this second chart, what we are comparing is the photo repair treatment with the UV treatment, because the UV treatment is the originally like my originally point of where I started this experiment. Okay, I used the UV and then I took these cells to the photo repair, and it's the same case for comparing dark repair and UV condition. So this is the UV proteome. We have 24 proteins that are downregulated and 14 proteins that are upregulated. When we compare photo repair with the condition of UV, we see that there are 32 proteins that are upregulated, and in the dark there are less proteins that are upregulating, and these are 23 proteins. Then we can compare the different conditions and compare if they were downregulated with the ones that were downregulating, upregulated, downregulated. So it's a little bit of a mess here, but I want to just simplify it to say that all the proteins that were upregulated in in UV, they were unique for this condition. Okay. So they were not over-regulated in photo repair or in dark repair. The ones that were inhibited or down-regulated in UV, some of them are over-regulated in photo repair and over-regulated in dark repair. And this is just, it's a little bit complex, but what we are trying to figure out is how all these proteins are switching on and switching off in these complex conditions. And what we are trying to do is to integrate all these proteins in a system like this that will be the UV proteome. So this is in the condition of UV, trying to integrate the 14 proteins that I showed you before. In this chart, the higher the blue, the higher the upregulation, and the higher the size of the circle of the figure is, the higher the value of this protein. Okay, and I will not go into detail of this, but just to mention that we have identified at least 2 different systems here, or group the proteins according to the function, and a set of proteins that are acting actively upregulated in the UV proteome or downregulated, are related, of course, with the metabolisms of the nucleic acids, so like the REC-A protein or the RNA polymerase or other components regulated the transcription. And the other group of proteins that we have put here are related with the stress response, so the catalase or all the metabolic pathway that give reduction equivalence to the system of the oxidative stress response to initiate or to protect the cell from the reactive oxygen species. And I want to call your attention to this protein, which is GBPK. This is the protein, and all of the ones that are here also, that are forming part of the operand of these gas vesicles. The one that is overexpressed is the regulator, GBPK. That means that under UV, the gas vesicle is being produced, and that may indicate that it has some kind of activity in the UV stress response, maybe for protection. And we are trying to integrate and characterize each of these proteins in each of the conditions. But to make it short, I want to give you a few messages so you can take home these ideas only. I want to highlight that the high-altitude Andean lakes are a rich source for UV-resistant Actinobacteria and other polystreamophiles, that especially Actinobacteria are interesting and unique models for studying all these molecular and physiological mechanisms that we call UV resistome, and that the UV resistome is complex and coordinated system of minor subsystems, but there are key proteins that maybe act as master regulators that drive all the changes, while most proteome stay similar among conditions. Here my question is that maybe because this strain is resistant, it has this behavior. We have to take some sensitive strains and see if they have, if they alter all the proteomes, or if they have key proteins, as in the case of Sterencone. So I want to thank all my group, PhD students, Federico, Natalia, Daniel, Silvina, postdocs, lab assistants in Center for Electron Microscopy. And of course, this work was made by first initiative of Dr. Farias in the PROIMI and LIMLA lab, making the sampling and the isolation of the strains and the DNA. For the climatology, we work together with Rubén Piacentini, researcher from Rosario in Argentina. For the photochemistry, we work with Claudio Borsarelli in Santiago del Estero. I made most of the work on the UV resistome and in the photoreceptors. I made as a postdoc, as a Marie Curie scholar in Professor Wolfgang Gartner lab in the Max Planck Institute for Chemical Energy Conversion. And we have the support and the experimental work of Thierry Duqui and Jean Cadet in Grenoble, that they help us with the determination of the photoproducts in the DNA. I want to take the possibility, also the chance to show you where we are. We are in the northwest of Argentina, in Tucumán. This is our institute, the Center for Electron Microscopy, which is quite new. It has 2 years. We have a very nice platform for electron microscopy, so transmission electron microscopy and scanning electron microscopy. We are a co-facility. That means that we do services in different fields, in medicine, in nanobiotechnology, in forensic and environmental science, in geology, in bioanalysis, and especially in my case, we have my group there in the part of microbial, molecular microbiology and ultrastructural microbiology. Apart from the strains from the lakes that I show you, we are working with other pristine wild environments near. We are in a rainforest area in Tucumán, so we have rainforest to sample and analyze also microbes from there. We also work with the environment, polluted environments. We have a lot of mining activities here, so we are sampling this. And recently we have started using the sampling the urban microbiome, and now we are using this approach to study, to making a monitoring of the SARS-CoV-2 also. So I want to invite you. I know now it's difficult to travel, but in the case you are interested in doing a postdoc, we are open to, there are many opportunities like the Marie Curie Global Fellowships. I will be very glad to be your host if you are interested. So this work was supported with the system of National Research System of CONICET. I'm a researcher from CONICET, the Agency for Science in Argentina, National University of Tucumán, the Ministry of Science. Well, I said that I have a Marie Curie Fellowship in Germany, DAD grants, and of course the support for the Max Planck Society when I was in the thank you, and I'm open to questions if there is time for it.
Brilliant. Thank you very much for that talk, Virginia. We already actually have a few questions. So the first is from Ankita Nag, who asks, what are the SOS repair genes expressed in the bacteria, and how do you compare this organism to Deinococcus radiodurans?
Okay, very nice question. Well, the SOS genes are here, well, but they are expressed in a condition, especially the polymerase that bypass the the damage. Okay, so we have a great number of proteins expressed for this, and with the Deinococcus, well, Deinococcus, the proteome and the genome that I have read about Deinococcus has to be more with the X-ray, like the radiation damage, the single strand breaks, and I think it's quite different. Deinococcus radiodurans also have this shield, special, a shield that is protecting a structure on the wall that we did not see in our strain. So this is, I will show you. This is a electron microscope, transmission electron microscopic imaging from Coniac 20, and it doesn't seem that it has this shield from the Deinococcus. I think these are the both difference. One is more single strand break oriented or to fix this, and the other, and also the shield.
Great, thank you. We have a question from Thomas McLean, who asks, do you think that the HALs such as ACT20 are reacting directly to the presence of UV or the results of that, e.g. DNA damage and increase in cellular free radicals, or a combination of both? And have you managed to find any such sensor systems?
Well, yes, this is very interesting. Actually, what we want to see if they can sense directly the UV, that would be very unique. And it for me is very obvious that it must have something that can sense. But we check with these UV R8 receptors that are in plants with the genome, but not only with this strain, but with other strains, and we cannot find the similarities. So maybe if they sense the UV, well, maybe they are sensing the UVA with the cryptochromes, and with the photolyase they can sense UVA. But it would be interesting to find a sensor to UVB. And we had to think, yes, it's hard to design a experiment to check the difference, if they sense directly or if they sense that there is a damage and that's why they respond. Yeah, this is the next step.
Great, thank you. From Beatrice Camara, she asks that there's a surprisingly small genome for Nesterinoconia. Did you see any BGCs?
BGCs?
Biosynthetic gene clusters.
Yes, these are the polypeptides. Polypeptides, chalcone synthetase. We found that there are a very rare biosynthesis of antibiotics, A-lipids, or something like this. Yes, that. But the most clear is the one for the chalcone synthase, which is a polypeptide pathway.
A related question from Alba Iglesias. She says, very nice presentation. Is Nesterinoconia living only in these kinds of extreme environments, or can it be found in other environments as well?
I saw that there were, most of them, there were allophiles. There was one Nesterinoconia which was isolated from the gut or from some human tissue. I think it was gut, which is quite extreme anyway. But no, in general, Nesterinoconia is from at least the one that I described.
And I think one final question in the interest of time from Thomas McLean. If you do know how these biosynthetic gene clusters look, are they more towards UV survival than antimicrobial warfare? As the environment is so harsh, there is not too much microbial competition.
I would say that if they produce this, they produce it for competing with the ones that are there. Because even if we think that there are not biodiverse, if you compare, there are quite much biodiversity. So there are a lot of microbes that are resistant to these conditions, but they have very few nutrients, very few nutrients or elements. So I think one way of the resistance must be to produce some secondary metabolite, and we have also start looking for it with some essays, but till now it's preliminary.
Right. Well, thank you very much again for that talk.
No, thank you.
I'll just.
Bye.
We'll just wrap up, just briefly reminding people to please submit abstracts if they're interested in giving flash talks for our next seminar on Thursday, the 25th of June. And we hope to see you all then.